Yes, good evening to all the participants. Welcome to the day 6 session on molecular talking analysis and virtual screening. Before stepping it on to today's session, can we recap the last session topics? So, what exactly did we discuss in the last session? Yes, so we discussed something on talking. In talking, we discussed on protein ligand talking. That is, and then we also discussed something about protein talking. These are also types of talking we generally do, and we have discussed the mechanisms of molecular talking. So actually, there are three theories of molecular talking, isn't it? So what are the three theories of molecular talking? Yeah, we have seen the methods also, isn't it? Different methods of talking. Lock and key, good. Like lock and key, Fisher proposed by Fisher, isn't it? Fisher lock and key. The second model, second and third, induce fit, good. And the third is the lock and key, induce fit. In lock and key, we can see fixed protein, fixed ligand. So that is it's not flexible. Yes, the last one is the conformational ensemble. So these are the three methods: lock and key, that is Fisher lock and key, and Coastland induced fit, and Bayong conformational ensemble model. Okay. So these are the three models of three theories of molecular talking. Okay. What are the two important parameters in molecular talking? If you see in molecular talking, the commercial software distinguishes with freeware from these two important parameters. What are that? Yes, Autodoc is a freeware tool of ligand protein docking. But important parameters are scoring algorithm. Okay, I mean scoring algorithm. You remember? So yesterday I just taken to that kind of a thing. Okay, search algorithm. So basically, if you see the docking, so a search algorithm and scoring function. Okay. So how we search the binding sites. Okay. So search scoring function. Of course, scoring function we are using an algorithm and search. Okay. So search I mean finding the binding sites. Good. So we are also finding the binding energy in Autodoc. And then so which type of docking is not possible based on rigidity and flexibility of ligand and protein? So actually, three possibilities are there in ligand and receptor based on rigidity and flexibility. So what is not possible? Do you understand my question? Actually, yesterday, yes, I think someone, yes, flexible protein and rigid ligand. So this docking is not possible. But remaining all the other possibilities are possible, like flexible protein, flexible ligand, rigid protein, rigid ligand. Okay, and even this flexible protein, yes, rigid protein and flexible ligand. So that is also possible. Only combination which is not possible is this flexible protein and rigid ligand. And so we also discussed about the chemical complementarity. Instead of simply fitting a ligand and receptor, so based on the contacts, hydrogen bond contacts, whether it is contacting with aliphatic region or it is contacting with aromatic ring, so the number of hydrogen bonds varies, including the ionic contacts, hydrophobic contacts, that can also increase the number of hydrogen bonds. So the number of hydrogen bonds can determine the strength of binding of ligand and the receptor. Of course, we measure through binding energy in kilocalorie per mole. So, however, we calculate the binding energy is dependent on these bonds also. And we also find so many physicochemical properties of the ligand and receptor, and also the ligand receptor complex. We consider all these three to determine the scoring function, to determine the binding energy in case of autodock. And okay, what are the types of autodock? So we have discussed types of autodock on yesterday, isn't it? I discussed four types of autodock on yesterday. So what are the four types of autodock we have discussed? That number one, PYRX, Python RX programming, PINA, Autodock PINA. We have also seen in Swiss Talk. In Swiss Talk, we have an Autodock PINA. Very, very good. Autodock 4.0. That's it. So these are the four different tools we discussed in Autodock. Of course, covalent Autodock. Covalent Autodock, which is missing, you have mentioned. Okay, so then what else did we discuss? Protein talking servers. Give me example for protein talking servers. And similarly, ligand protein. First, protein is got 3D dock suit, haddock, patch dock, hex dock, plus pro. Good. Okay, so these are some of the protein talking tools. Especially, we have also seen there is a commercial program called Gold, isn't it? From Schrodinger, we have Gold to do protein talking. Okay, and we have also seen the ligand receptor, I mean small molecule ligand receptor topic. Now today we'll be discussing on whatever we have terminated on yesterday. You remember on yesterday we are discussing something on how to do a manual docking using autodock. That is doing a manual docking of small molecule with a protein. The small molecule what we have chosen is a curcumin molecule. So we have chosen is curcumin molecule. So this is a curcumin molecule. Initially, we have downloaded the curcumin molecule in the form of STF format from PubChem. Later, we have converted this as .pdb from Discovery Studio Visualizer. And then whatever protein we had, protease, this I have docked. I docked curcumin with the protease protein. And finally, before closing it, okay, so after completing 5 steps, I have saved this complex file as curcumin underscore protease.pdb. So this is the final file uh, we we have saved it on yesterday. Curcumin underscore protease, that is curcumin protease complex.pdb. But before preparation, I told you we will first have a kind of a folder. In that folder, have autodoc4.exe, autogrid4.exe, and then have a tutorial. How you can get autodoc4 and autogrid4? For this, we should have MGL tools. So, can the last question I like to ask, because many of them, if you didn't try with Autodoc, you should know what are the tools, what are the softwares to install to work with Autodoc. Okay, what are the softwares needed to install Autodoc? Chimera, oh, Chimera CHIMER. Okay, so is very good. CHIMERA. So number one is Chimera. Next is EduPymol. Good. So EduPymol, it is educational tool, Python molecular wear. MGL tools, good. From scripts.edu, you can download the MGL tools. One thing is missing. Chimera, EduPymol, MGL tools. What is the next thing? No, not Pymol. Not. Uh, Autodoc suit, exactly. Autodoc suit. Okay. So these are the tools. In Google, you have to simply search for this. Autodoc suit, MGL tools. Okay. Like today, I'll be providing you a demo on Pyrex. If you want to download a PYRX, simply we have to type Autodoc PYRX in Google. It take you to the pyrx.sourceforge.io. And I believe today also I pasted few links. So if you click that link, it will directly take you to that. And MGL tools, if you type, okay, take to the scripts research, MGL tools. So whatever is there, 1.5.7, you can download. And for downloading the camera, simply camera download. Okay, so if you type the camera download, download UCSF camera. So this is the URL we have to use. Okay, so whatever is meant, because I will, whatever you find in this chat, okay, you can also follow this. Okay, so now let me go ahead with the continuation of what we completed on yesterday. So we, whatever we got as a complex file, I'm going to open with Discovery Studio Visualizer. Again, for your information, this BioVia Discovery Studio 2023 is a free visualizer. Okay, so even we can create peptides using this. See, we have built an edit protein. If you want to create a peptide, you can use this amino acid buttons and you can create it. We have plenty of options to work even with the freeware of BioVia Discovery Studio Visualizer. In 2014, the Asteris company who owned this Discovery Studio has sold to BioVia of Japan. Asteris is from USA company and BioVia is from Japan company. Now this is a product of a Japan company, BioVia. So see this, this is our protein. Okay, what protein? Protease. And whatever you see this ligand, okay, is in stick model. So the ligand is in stick model and this protein is in solid ribbon model. So if want to do the analysis, I suggest you first to go to view and then click hierarchy. If you click view hierarchy, you can able to see the details of the atoms. So here in this case, A chain protein. Okay, you can see the amino acids of the protein. And in this chain, we had something. So here we have UNK0. The UNK0 stands for the ligand, whatever we have talked. So these are the parameters. Okay, these are the coordinates. Whatever coordinates we find, we can able to see it in view hierarchy option. Okay, the next to analyze the results, I suggest you to use first receptor ligand interactions. In the receptor ligand interactions, if you click this, you get show 2D diagram. If you click this show 2D diagram, you can able to see this 2D diagram interaction. So directly you can place wherever you want to place and we can zoom it. Okay. So also these are needed actually. The bottom thing, whatever mentioned as interactions, you can see what interactions, van der Waals, conventional hydrogen bond, pi sigma, pi sulfur, alkyl and pi alkyl. So these are the bonds of interaction of ligand and receptor. And if you see these hydrogen bonds are shown in green color, the dark green color or conventional hydrogen bonds. Okay, so now if you want to get a kind of an output, okay, a decent kind of an output to be published in reputed journals, so better you can also change any kind of a style. For example, if I go with the disk instead of line, or if I go with a stick, earlier it was in line model of a ligand. Now I am changing it to a stick model. So see, this ligand alone has changed to a stick model from line model. And residue, this denotes the amino acid contacts. So if you want to change contact, I mean this display style from ball to disk, see this disk is appeared more contrast. So if you want to change any kind of interactions, through display, you can able to change the display here. So now if you want to go further with an interaction menu, we can able to see hydrogen bonds, which are strong bonds between the ligand and receptor. And we can also find electrostatic, hydrophobic, halogen bonds, miscellaneous bonds, unfavorable bonds. So like this, we have types of bonds in this interaction. Okay, and if I want to show only the hydrogen bonds, and if I don't want the other bonds, you can also deselect the bonds. Okay, whatever you don't want, we can also deselect the bonds. So whatever earlier we had, I deselected. Okay, so whatever I deselected now, so I'm going to click show distance, apply. See, we can able to get the hydrogen bond length now. So I click this show distance. We got the length as 3.51. That is this, the red color, whatever you see in the ligand is an oxygen moiety. So this is actually double bond O of a ligand is interacting with the asparagine 98. And this is the hydrogen bond length, that is 3.51 angstrom. And similarly, the leucine of fifth position, and A denotes for the chain. There is only one chain in this protein. Okay, it's interacting with this OH group. So H is above and this is O. Okay, the H is generally shown like this. So you can able to see it's a polar kind of polar compound interacting with definitely a polar functional groups. Okay, so now let me go ahead with the 3D interaction. Till now, whatever we have seen is the 2D interaction. So if you want to see the description of the bonds of asparagine 98 and leucine 5, which is not shown here, then what else we have? Asparagine 98, leucine 5, 24. So 5, 24, 98. So these are the positions. 5, 24, and 28. Okay. So whatever we have seen the positions of an amino acids, we can't able to see the atoms of the amino acids. If you want to check the atoms, so I suggest you to go see in this 3D viewer. So whatever I shown you is the 2D diagram. Now this is your 3D diagram. In this 3D diagram, so after selecting this, select the scripts, selection, select ligands. If you click the select ligands, only the ligand alone gets selected. Next, scripts selection, select atoms within 4 angstrom. Selection, select atoms within 4 angstrom. So whatever atoms near to the 4 atoms, that is selected at present. The next third step is scripts, ligand interactions, show ligand binding site atoms. That's it. Only 3 steps. Again, I tell you, first scripts, selection, select ligands. Next, select atoms within 4 angstrom. The next, go to the ligand interactions, show ligand binding site atoms. So that's it. Now we got this kind of hydrogen bond monitor. In the hydrogen bond monitor, we can able to see this coordinates. Okay, leucine 5 of HN, that is amino acid contact atom, is visible now. Leucine 5, HN of leucine 5 is interacting with the oxygen moiety of the ligand. And 96, 309, not 96, 98 asparagine is interacting with the ligand. The second interaction is amino acid of a protein with amino acid of a protein. This is not considered as a ligand protein interaction. This is a protein protein interaction. We have only 3 ligand protein interactions. One is interaction with leucine 5, the other is asparagine 98, another is leucine 24. Okay, so these 3 interactions, and you can also see the atom-wise interaction in the amino acid of a protein as well as the atom-wise interaction in the ligand. Okay, this is also one of the data. One data is taking a 2D diagram, the other data is this hydrogen bond interactions, including that hydrogen bond length, etc., whatever I have shown. And the third is to take even a 3D diagram. Either 3D diagram or 2D diagram can be shown for the publication. So it can decide. But this 3D diagram not looks nice. How to make it nice? So can we go ahead with changing the display patterns and even show highlighting the amino acids specifically, like 5th, 24th, and 98. Okay, I'm going to do that now. So first the only that selected binding site amino acids are shown. So I deselect everything and I select everything. So the entire protein came once again. So the binding amino acids are 5th amino acid, 24th amino acid, and 28th amino acid. Only these 3 alone I am going to select. Am I right? 5, 5, 24, 98. Not 28, 98. So I have pressed control and I am just pressing this. So only these three amino acids got selected, whatever binding sites we have determined in that hydrogen bond monitor. Later, I go to the structures, labels, add. Inside the labels, I'm going to keep it as amino acid, and in amino acid, I'll be selecting the 3-letter insertion code, and I'm going to increase the font. I increase it to a large font. So now I got an amino acid labels. After getting these amino acid labels, I right click here, go to the display style. So whatever amino acids we have selected, still the protein can be in solid ribbon, but the atom, whatever we find in line, I want to make it as ball and stick. So this is the display I'm changing for the amino acids, the selected amino acids. Click apply. So now ball and stick model is there. That is binding site amino acids are changed to ball and stick. But the ligand is present in stick model. The entire protein is present in solid ribbon model. So it looks nice, okay. And but for the publication, you should not have black background. So go to the graphics, instead of background color, black, click this white, and now apply. Now see how it looks like. So this is the interaction patterns. So while taking a snapshot, okay, so we can, uh, so while taking the snapshot, we can can maximize this, okay, and even if you want to remove the hierarchy, remove the hierarchy. So now we can, if you drag and see, you can even see the hydrogen bond interactions. So that the green dots are visible now, the green dots. So take a snapshot. So even this, if you want to change the font color, you can change the font color of the ligands. So this is also one of the output of the docking analysis. Now let me go ahead with the today's session. So today's session will be discussing on, of course, docking analysis we have covered. The next thing what we discuss is the virtual screening. So the molecular docking analysis by which Discovery Studio is completed already. So today we discuss a few more topics like virtual screening, lead hopping, high throughput screening, virtual screening methods. We have methods like ligand based, a structure based, and the third is the hybrid based. And virtual screening softwares, we see examples like Farmer, Pharmagist, Ligand Scout, Tovis, Autodoc PVRX. Today we practice Autodoc PVRX. Okay, so this, now let me go ahead with the journal publications. I believe I have sent these journal publications on yesterday. Just go through that, how the docking is done, how the results are interpreted. Today also I have circulated one more journal paper. So you can go through how the docking results are tabulated in that. Very simple experiments. Okay, for publishing a paper, I'm doing a research for a single day, and that data is sufficient enough to publish in the journal paper. So only thing is that what exactly to take, what not to take. Okay, so the determination, I mean, deciding thing is important for publications. Now let me go ahead with this first publication. I told you on yesterday itself that a plant compound vitafrin A, which is taken from ashwagandha, I have targeted two important receptors of cardiovascular diseases. Here I have used molecular docking and AMD simulation. So if you see in this molecular docking, other than taking the two receptors, I've also taken 7 more receptors, totally 9 important target receptors responsible for cardiovascular diseases that I have taken. Okay, so that corresponding PDB IDs are mentioned here, 9 PDB IDs. So initially I tried with a few standard compounds also. For example, for mineralocorticoid receptor, aldosterone is the standard compound. Okay, standard drug, something like that. So I just taken the standard compounds and I docked it and I compared with the vitafrin A. So if you see this vitafrin A, which is abbreviated as WFA, has also shown good scores with the MAFP, PAFH, PRP1, and this also showing best binding affinity with P2Y1, and similarly. Uh, SCD, but this is not significant. I told you, orthodox score, if you get -6 and below, it is significant. So even this is not significant. Yeah, Vitafrin is significant. Vitafrin is significant, but the standard compound, whatever is selected, is not significant. Okay, so the Vitafrin has shown good results for this. Okay, for these two receptors. Okay, and I just taken these receptors, whatever docked well, and then taken for MD simulation. Okay, so the interaction energies are shown, and similarly, we have a tool for calculating DSX scores. So DSX scores are also shown. So these two kind of results, the DSX scores something related with SAS potentials, and binding energy in orthodox is represented in kilocalorie per mole. So this is actually a kind of interacting atoms in amino acid, and this is an interacting atom of the ligand. And I mean, especially if you see in the ligand, we have Ca C8 of WFA. Okay, and these are atoms of that standard compounds. And then you can see the length of bonds, etc. So case study 2. Molecular docking and simulation, the similar kind of studies, but with a different compound called pyrivinone with the two receptors of hepatocellular carcinoma. So here, if you see the docking results or like binding energy is represented in terms of kilocalorie per mole. Okay, whatever docked with the BEB, you can see almost all the results we got above, I mean below minus 0.6. So whatever we talk with BB has generated a significant result. Okay, and also comparatively, it is showing good score when compared with multikinase inhibitor sorafenib. You know, sorafenib is the popular drug uh, used for treatment of hepatocellular carcinoma up to the third stage. This is a popular chemotherapy drug actually. But when compared to this sorafenib, which is targeting multiple receptors, our plant compound is also showing a significant result. And these are the contacting amino acids. And case study 3. This are here also the ovarian cancer article. I just talked with three important plant compounds. Okay, that is epigenin, quercetin, resveratrol, with the top 5 receptors of ovarian cancer. So if you see in this result, okay, the quercetin is showing good result here. That is FGFR1, number 2, number 3. Okay, 3 is NCBP1, number 4, the red is quercetin. Okay, number 5, RAC1, and number 6. So in the 6 receptors, out of 10 receptors, we find quercetin is showing a negative maximum energy. So just because it's showing a lead, we concluded the quercetin is the best compound for treatment of ovarian cancer. So, and then if you see, so till now we have discussed on how to interpolate the docking results. Now I'll be discussing something on virtual screening and types of virtual screening. So I told you on yesterday itself, in virtual screening, instead of docking a single ligand with the receptor, we try with multiple ligands. That is ligand 1, ligand 2. Okay, so I think better I can show it bottom. Ligand 2, ligand 3. Like this, we can dock up to ligand n. Okay, so up to this we can able to dock. So whatever we talk, okay, so whichever is showing the negative maximum energy, that we screen it as a best ligand. So how then we can compare these three virtual screening, lead hopping, increasing high throughput screening. This will be seen in next slides. First we discuss on virtual screening. So if you come across virtual screening, as I mentioned, multiple ligands are docked simultaneously with the target receptor at the same time. So you can see whatever we do with this protocol, okay, it shows less accuracy. If you see, this is The y-axis stands for accuracy, and the x-axis stands for the number of molecules. A single ligand and a single receptor docking is somewhat more accurate, okay, when compared to docking multiple ligands with a receptor. So generally, whatever we do with Autodoc PVRX, that is used as one of the screening protocol, and whatever you get like the top two drugs or top three drugs, we can also do a manual docking later. Okay, so visual screening is preliminary step of the manual Autodoc. So first we have to screen this. Why we are doing this preliminary screening, here we just can get the binding energy, and even whatever, if you want to analyze the hydrogen bond contacts and etc. So the better you can go with this manual talking using Autodoc. The next type of virtual screening is lead hopping. In lead hopping, we initially start with initial lead. So whichever is marketed, whichever is in the market for treatment of a disease, okay, we start with it. If we start with that kind of a research, okay, that is a kind of initial lead we take. So whatever we take initial lead, we all might have we have talked with the receptor also for the particular biological effect. But if you want to improve the efficacy of the existing drug, I suggest you to go with fragment-based screening. Fragment-based screening. In fragment-based screening, which is also called as lead hopping, so we cut the uh, ligand, that lead, in different fragments. Whatever we cut into and we generated different fragments. Okay, so we try to link with some linkers and spaces. Here, see, this structure is something difficult. The first structure, whatever you see, is difficult, and now whatever we cut into multiple fragments and whatever we assemble with the linkers and spaces, that structure is different. So this is a new lead now. So if this new lead can generate, for example, if this shows minus 8 kilocalorie per mole with a particular target, and if my new lead can give minus 9 kilocalorie per mole, then this is the best lead whatever we have generated. So with the existing drugs, we can also develop new lead compounds through fragment-based screening, and this process is what we call it as the lead hopping. Okay, and the third one is the increasing high throughput screening rates. For increasing high throughput screening hit rates, okay, so we can take an example like whatever we have completed in IGM 2016. In IGM 2016 of IGM SVC 2016, that is International Genetically Engineered Machines Competition, which is conducted by MIT USA. I mentored a team of students under computational studies. So in their studies, they have taken an example like the project is actually Lactose Shield. The project is Lactose Shield done by the SVC students, and they secured the silver medal. So that is the first silver medal secured by IGM team of SVC students. In that Lactose Shield project, our objective is to protect the milk. That is, if you want to preserve milk for a longer duration, okay. So how to improve the shell life of the milk? So that kind of a project we have named as Lactose Shield. Here in this, to improve the shell life, before going to the shell life of milk, like in Tamil Nadu, the milk packets from government, what we get is Avin, isn't it? If that is Avin in Tamil Nadu, Nandini in Karnataka, isn't it? So, what is the shell life of this packet milk, whatever we procure? This is my question. What is the shell life of packed milk? So I'm giving an example for high throughput screening. So 2 days, very good, excellent. That's the answer. Only 2 days, and the two for the 2 days we have to preserve in 4 degrees centigrades, isn't it? So And if you want to increase the shell life, you can also keep it in minus 20 degrees centigrade. It can we can get up to 7 days also. But if we can able to heat, even that can able to preserve up to 5 to 7 days. So this is the normal shell life of the packed milk. It can be Nandini milk or Arogya milk or the Avin milk, whatever we procure. So, but under the Lacto Shield project, so we can improve the shell life of the beer, the milk up to 3 to 4 months. Did you heard about something called ultra packed milk? Ultra packed milk. This you might have found in the in the pandemic time when we face the COVID-19 pandemic times. Okay, even the consumers also are procuring this ultra packed milk. Whatever we procure ultra packed milk, generally the 5 ml, 500 ml of milk, whatever we procure, the Avin is around 20 rupees, 20 to 30 rupees. But the ultra packed milk, the 500 ml, that is half liter, can come around even 45 to 60 rupees. So that is somewhat expensive, but we can able to preserve even up to 3 to 4 months in the fridge. Okay, so but similar kind of a project, okay, Lacto Shield. Here, our objective is to uh, protect protect from microbial pathogens. Okay, microbial contaminants. Okay, we just designed a project with constructing a 9 amino acid peptide. A 9 amino acid peptide, the tail region, if we have a tryptophan, okay, and we should have a 6 amino acid, and followed by the last three letters should be a tryptophan. Okay, so then what exactly you can find in this first amino acid space? Here in this first amino acid, you can have 1 to 20 amino acids in combination. Okay, and similarly in the second missed and the third, fourth, fifth. So we have generated a combinatorial peptide compounds, combinatorial list of peptide compounds. Whatever we generated, thousands of compounds according to this hypothesis, which can able to protect milk from the contaminants, microbial contaminants. Okay, we have generated more than 1,000 peptides, and we have talked simultaneously with a single target. So whatever we talked, okay, so this kind of an process is what we call it as increasing high throughput screening. So even in internet, if you search for IGM 2000 2016 SVC lactose shield, you can find the description of that project. Now, next is flowchart of virtual screening. So virtual screening, can also be classified as a ligand based and structure based. In structure based, if you go with the protein ligand docking, so generally we go like this. The structure based is a ligand protein docking. But in ligand based, if you have actives, that is, if you already this compounds are existing in the market, and there are also something called inactives. These inactives are a synthetic compounds, not yet synthesized, not yet present in the market, but later, if the chemistry people, the medicinal chemistry, can also bring this kind of a compounds based on the computational studies and also the feasibility of developing these compounds. Okay, so if you have an actives, we can also go with the similarity searching, and we can also go with pharmacophore mapping. And if you have both actives and inactives, we can try with the machine learning. So when we can try with the machine learning, so our the theme of the internship course is machine learning, isn't it? We have already plenty of tools for talking and plenty of tools for simulation, pharmacophore mapping, etc. Then when to go with this kind of a machine learning to develop a new tool? That's most important. If you have inactives, then we can try to come up with a kind of a machine learning approach for coming up with any kind of a drug design tool, drug design method tool. Now let me come across the types of virtual screening methods in drug discovery. So that we simply restricted as ligand based and structure based, but we also have a third type called hybrid based. Under ligand based, the best example is pharmacophore modeling, QACR. okay, where you need only the 3D structure of the ligand. The 3D structure of a ligand is sufficient enough to analyze the to even screen multiple compounds from a single compound. If you want to screen which is the best lead, you can use these methods through a 3D structure of a ligand. But if only if you have a 3D structure of a ligand and 3D structure of a receptor, both are needed. If only if you need have these structures, then only we can call it as a structure-based screening. So structure-based screening, we can use molecular docking as well as MD simulation. In hybrid-based, either ligand or or ligand and receptor. Okay, ligand and receptor. So you must have either 3D structure of ligand or 3D structure of ligand and receptor. If you have like this is what we call it as hybrid based. The example is a pharmacophore based docking. Okay. So in pharmacophore can also be linked with docking. Under that case, we call it as hybrid based. Now we discuss the virtual screening softwares. Virtual screening softwares, Pharmagist, PVRX, Ligand Scouter, Dovis. So these are some of the tools, and you see the Dovis. Many of the online tools are also using the Autodoc. Even yesterday we have seen the Swiss doc, which is deploying the Autodoc Mina, and Dovis is using Autodoc 4.0, and PVRX is what we will be discussing it on today. Now let me go with the Autodoc PVRX. So Autodoc PVRX, the name itself implies Python RX programming. So that's the expansion of the PVRX, and this is used. This is a freeware. So simply, if you go with the pvrx.sourceforce.io, we can able to download a single file, a single exe file, whatever we download from this URL, which I have shown here. This we can install, and after the installation, we can see the window like this. Okay, a single software is sufficient enough for working with Autodoc PVRX. And for working with the PVRX, of course, we need a subset of compounds, okay, not a single drug. So to download a multiple compounds, we can use databases like ChemSpider, Drug Bank, CHEMBL. So these are even KEGG, CHEBI, NCPA PubChem, of course. We can also use human metabolome database. In human metabolome database, we can see the metabolites. I told you in Ashwagandha we have 41 methanoids. Okay, if you search in PubChem, you may have this 41. But in this human metabolome database, you can see around 5 methanoids. Okay, admit this is also a kind of a virtual screening. So if you have hundreds of compounds, and if you want to screen the best top 10 compounds, we can use this as a filter, virtual screening filter. Even trying with multiple compounds and screening the best 10 compounds, something like that, is also called as a virtual screening. So I'm showing you this. Now, this is an output from SwissDoc. Yesterday I showed you, no? So in the same Swiss ADMA, we also have SwissDoc. If you click that SwissDoc, okay, it asks you to upload the 3D structure of ligand as well as the 3D structure of the receptor. After uploading, and if you click submit, you get this kind of a result. The result what we get is estimated change in Gibbs free energy. You can see from top ranked pose to the declining. In the descending order, you can see the different poses of contact of ligand and receptor. So in this example, I talked without a with a protease, I believe. Okay, and this is another database data set for downloading the compounds, Enamine. In Enamine, we have different kind of libraries, AI-enabled libraries, diverse libraries, targeted libraries. Libraries, we mean a set of compounds. If you see here, this high fidelity fragment library, so where we can use the fragment-based screening, 1920 compounds are available. Okay, like this, you can see DSI, disease specificity index, poised library, 860 compounds are available. So like this, in each and every library, we have some set of compounds. We can download that set of, for example, if I want to try with a high fidelity fragment library, click this download file option present here. So it can download all the 1920 in a single file. That single file, you can take it as a ligand file, and it can whatever receptor file like a protease.pdb, whatever I showed you on yesterday, you have a single protein and you can try to dock with it. Okay, so like this we can try with downloading the chemical data sets and for PVRX installation, this is the default thing we use. Okay, now let me go ahead with the hands-on session with the Autodoc PVRX. So till now we discussed on how to install and also how to install ASD Chem Sketch. To install ASD Chem Sketch in Google, type ASD Chem Sketch, it directs you to the asdlabs.com. Download ChemSketch freeware. Okay, if you want to work with compounds, similar compounds, you can also use this kind of a software tool. Now let me go ahead with the PYRX. So for PYRX, we can prepare a kind of a folder. Okay, and then um, we can try. So first I am going to prepare a folder called PYRX underscore demo 4. Okay, I just created like this. And yesterday we have one protein called protease. I'm going to take this because it's the smallest protein to work. So I'm going to try with this protease protein. And then what exactly we want is the ligand. To download the ligand, I'm going to take you to the um PubChem. Okay, that's easy to work. NCBI PubChem. So if you go to the PubChem, okay, so we can able to can I try with a compound called pyrivinone. So pyrivinone or rotinoid compounds, isoflavone compounds from Pyrevia diffusa. From Pyrevia diffusa. Also this Pyrevia diffusa we call it as Punarnava. So blood rejuvenation. This is the best use for treatment of kidney diseases, kidney and liver diseases. If I typed pyrivinone, I got 15 similar compounds like pyrivinone B, pyrivinone G, E, so that kind of H. Like this, we have 15 compounds. Now, I don't like to talk all the 15 compounds. I want to screen initially. So, if you want to screen initially, if I click this download, I'll be getting all these 15. That doesn't make that all the 15 will be downloaded. If only if you have a 3D structure, see if I click this download, you have an options like download 2D, 3D. If you click 3D, whatever 3D structures available out of 15, that only will be downloaded as a single file. For example, only 12 structures have 3D, then whatever you have selected, only the 12 out of 15 will be getting downloaded. But I don't want even to talk all the 12 compounds. I want to filter further. To filter the compounds based on the physicochemical properties, okay, I just go with the filter. I just used a refine filter taken to the window. Yes. Now I'm going to filter this. I've changed the window. Earlier it was not like that. Earlier it was very easy to do that. Now we have to remember everything. So even whatever we filter, they have given filter by date. So what actually in NCBA, you can register and you can log in. Whatever you work, it will be there in our history. You can search in search history. So they just implemented it, I believe. Okay, whatever we are searching, they just implemented. So if I want to screen hydrogen bond donors from 3 to 4, okay, so this I'm using it as one of the filter. So after filtering, I got 11 results. Earlier I got 15, now only 11 results. Now whatever I got, I just used a physicochemical property that is changing this parameter, so you can change the parameter. Now I want to download only 3D. So I click this 3D. Compression, you keep it as none or it will be chipped as zip file. So now whatever I have this, okay, a 3D 11 compounds, I'm going to download. So click download. So what happened? Download. I just zoom out and see. Still I have to zoom out. Download in what format? STF format. So we have to click the STF. So now that 11 compounds related thing I'll be downloading in this PVRX underscore demo. So this is related to PyRV known. Okay, whatever I download the PyRV known is in the form of STF format at present. Okay, whatever I downloaded is in STF. Now just open this STF and let us see how many compounds are there in this PyRV known. So I closed the earlier windows. Curcumin protease I closed. Okay, save as no. See, only 10 compounds are downloaded. It means that the 11th compound don't have 3D structure. So if you want to see all these compounds in the visualizer, select all the compounds. See, the 10 compounds are present in a single file. Okay, it's not a single compound, multiple compounds. Now let us see how to work with the Autodoc PVRX. So the preparation of ligand and receptor is completed. Now for after installing this PVRX, if you type PVRX, you get an PVRX app. You're opening this app. So initially, like however we have set the path for Autodoc. Even in PVRX, we have to initially set the path, and very few steps we have to make it. Unlike Autodoc, manual docking using Autodoc, we use plenty of steps and it took much time. That to here now at present, I'm going to talk all the 10 ligands with a receptor in a short period of time. Okay, so I suggest you to first try with this virtual screening, which is somewhat less accurate. Later, you can try with the screened compounds to do the manual autodoc. Now, the first step is to set the path. So, to set the path, file edit preferences. So, in the file edit preferences, the one whatever I kept is the PVRX demo 4. So, I just uh, saving my workspace here. So whatever intermediate files I'm to like to generate, I'm going to generate in this workspace. Or simply click the browse and select wherever it is. This is pvrx underscore demo 4. Okay. So click OK. Now setting the path is completed in edit preferences. The next step is go to the file load molecule. In the load molecule, we have to locate to the desktop. In the desktop, we have pvrx demo. Protease is our file. That is protein file. Click open. So whatever protein we open, we can able to see here. This is also a kind of a Python molecular viewer. So if you see this protein, okay, protease, I am going to right click initially Autodoc make macromolecule. This is the next step. After loading the protein, Autodoc make macromolecule. So now the protein, what has happened? The protease is converted as protease.pdbqt. Earlier it was not converted. I told you if you want to talk a ligand with a receptor, all the files initially we have to convert. That is all the files I mean the ligand and protein files has to be converted to pdbqt. Then only we can go for talking. And in the Autodoc initially we develop the grid. Okay, after setting the grid only we go for talking. So the same step here also we make. Now I just the first step I converted the protease.pdb as pdbqt. Okay, in a single step. Now the next step is to go to this Vina wizard. Okay, in the Vina wizard, no, instead of Vina wizard, better go with Open Babel. The purpose of going to the Open Babel is whatever I downloaded the ligand, very known, is .stf format. But the Autodoc can accept only the PDBQT format. So we have to convert that file format to this file called format, PDBQT format. So I go to the Open Babel, which is inbuilt in this Autodoc PVRX itself. So after going to the Open Babel in the bottom, there is a green color icon showing insert new item. Click this. So now we have to locate to the desktop demo, PVRX demo. Now I'm going to select the query window. So whatever ligands I have selected, that 10 ligands are shown now. I mean, the 10 ligands first you have to select by clicking shift and the first and last. If you continuously press shift and you are selecting the first and last, all the 10 are got selected now. So later If you want to do anything, you can do, or you simply, if you want to do with a single thing also, it's possible. Just select a single ligand. The next step, what we have to make is minimize all. The minimize all, this button can direct you to calculate the energy minimization. Okay, the energy minimization is important to check how stable a ligand is going to bind with the protein. So to do the energy minimization for all the ligands, whatever we have opened, I'm going to do with minimize all. If I want to do energy minimization for a single ligand, I can use minimize selected. But our purpose is to do with all the ligands. So minimize all. So all the ligands are calculated with energy. Okay. The next step is go with convert all autotalk ligand PDBQT instead of going selecting a particular ligand. Now I'm converting all the 10 ligands as PDBQT format. So this step is also completed. So in this step, you know what has happened. All the ligands molecules are converted as PDBQT, which you can find here. All the 10 ligands. Earlier this step was not completed. Now only I have completed in Open Babel. Now our purpose is go to the Vina wizard. So we are going to the Vina wizard. Now click start. In this start, if you click, you see the protein molecule protease.pdbqt is selected, but no ligand selected. We have to select all the ligands for talking. So I am selecting here the first ligand and selecting the last ligand. So if you press shift and select first and last, you get 10 ligands selected, and protein is already selected. So click forward. So now we can get a kind of a grid box, whatever grid box you got. Okay. So if you see in this isometric view, a cubical kind of a grid box you can see now after clicking the cube. Now we just have to increase the size. Our objective is to not miss any of the binding sites. Okay. So we are enclosing our protein in this grid box. We are enclosing the to our protein in this grid box. So we are stretching the dimensions and we are extending the to keep. So how much large a grid box is there? That much time it takes for talking. Now I fit this protein in this grid box. The next step is the last step. After completing the grid, simply click forward. That's it. Now the docking is getting happened. All the 10 ligands are simultaneously docking with the protease protein present. The first ligand is already docked. We got the result, finished. Second ligand is also docked. Now it's going for the third, fourth. So it is a rapid generation. You can generate the binding energies of all these ligands within a short period of time. Okay. So first, this is a preliminary step. First, complete the virtual screening with PYRX. Then we can go with manual autodoc. So after this, I mentioned you like how to analyze these results. So we'll be getting results within a few minutes. And tomorrow also we have the session. And tomorrow we have on energy minimization and molecular dynamics simulation. Okay. So up to on tomorrow we'll be completing the target discovery and design and the recent contemporary researchers in drug design on systems biology approach. So in system biology approach also take you to the MATLAB, how to use the MATLAB, etc. Okay, so also I'll be teaching you that. Good. We have generated the results. Whatever results we have generated, okay, we have an option here, save as comma separated value files. Click this. Comma separated value files can be opened in Excel. So I go to the desktop and go to the PVRX underscore demo, and this is actually where we know talked with the protease, isn't it? So I save it like this. Okay. So Voirevinon underscore protease dot csv. If this is completed, you can close the window. Okay, because everything is done. Even the file, whatever output you want to save, we have saved it. Now I just open the We can see this very known underscore protease. So we can see this kind of results. Okay, so we can see RMSD0, it means that it is a base pose. All the 10 ligands are docked with 10 poses. For example, if you see this, okay, this ligand, this is the first ligand. If you want to know, if you type this ID in NCP PubChem, you can get the name of the compound. Okay, so like this, you have a series of. This is actually a first ligand result. So this is actually first ligand result. We got the 9 best conformers, and this for the second ligand. So you can see 1153. Okay, so for this second ligand also you'll be getting the 9 poses. The top one is the best top pose. So only we are getting the negative maximum. The remaining are all different conformers with higher scores. So if you want to screen which is the best ligand, I suggest you to select this binding affinity and go with data filter. In the filter, you can see which is the maximum, minus 8.6 is the maximum. You select this minus 8.6. Only one ligand has shown this minus 8.6 in the first pose. So this is the PubChem ID. Select this PubChem ID. And now I'm going to search that. I go to the PubChem and then I'm going to type that PubChem ID, whatever is searched. It is showing like boirevinon I. Boirevinon I is docked well with our protease protein and has shown the score around how much the binding affinity score is around minus 8.6. That's what we can able to find is minus 8.6. Okay, this is how we have to conclude. Okay, so I have completed today's session. Any doubts you can ask me. Sir, in this Autodock Pyrex, can we dock the newly synthesized drugs? Yes, yes, yes. Of course, superb question. Actually, there are also few more databases. I was explaining you on yesterday also, isn't it? We have databases like Zinc15. Zinc15 is actually a synthetic molecule database. This is what I mentioned you, that even in University of California, one scientist has docked in the supercomputer with 1.3 billion compounds. See, this Zinc15 is a free database of commercially available compounds as well as the synthetic compounds, purchasable, non-purchasable, everything is there. To go to the compounds present, click tranches. In the tranches, you can see the compounds are sorted based on the physicochemical properties like molecular weight versus log P. So if you want to download a set of compounds which is going to have molecular weight of around 350 and you want to get a log partition coefficient of 2.5, you have to select this. A molecular weight of 350 and log partition coefficient of 2.5. 41 million compounds are available. 41 million compounds out of 957 million compounds. Okay, and whatever you download, we download the 41. So this is for 2D, but generally for docking, we have to go with the 3D. The same property, if you want to check with the 3D, select the 3D, 350 and 2.5 select, you download, you select that, and there is a download option. Click the download option, you download all the 3D compounds. Like however we have done with Autodock PVRX, we can do the same. But for the large number of data sets, the 350 and 2.5, we again have 48 million compounds. Whatever we have selected, we have to use this option for download at the top. Okay, like this, we have even synthetic base databases, Zinc15, Zinc20, etc. So Autodock PVRX can be used, but you should have a high configuration server. If the generally our laptops, the i5 laptops can support around to dock 50 compounds at a time. i7 can extend up to 60 or 75, something like that. But if you want to talk up to the millions of compounds, you need a workstation or a server. Okay, sometimes you need a supercomputer to complete this task if you have large numbers. Any other doubts you have? The input files, no need to synthesize. We have plenty of input files, okay, in various databases. Just like that, you have to.
Uh,
Yeah.
No, sir. Actually, I asked like if our synthesis group is synthesizing any new drug, so that organic compound, that data won't be in that data bank, na. It will be with us only, na.
If
So,
so, so
okay,
whatever we get the coordinates, okay, whatever we got the coordinates, first we have to open the notepad and we have to edit the coordinates. So whatever I just show file new tab. So I just no, yes. See, for each and every compound, so yeah, the first compound start from here and complete from here. The second compound start from here. So simply like that, whatever we have multiple single compounds, paste it in the same file. Paste
okay,
it in the same file so that multiple coordinates will be in the single file, and you can talk all the all the ligands simultaneously with the receptor like this. So only thing is that how to prepare the ligand file in that case. Okay. Any other doubts? If no doubts, thank you very much for your interaction and patience. And also, I thank especially you joined even in the holiday. So tomorrow also we try to complete a most important session in MD simulation. Have a great day to you all. Bye.